Dear students, today we are going to discuss something about chick embryos, whole mount of chick embryos, particularly 18 hours chick embryo, 24 hours chick embryo, 36 hours chick embryo, and 48 hours chick embryo. First one, whole mount of 18 hours chick embryo. The embryo has central transparent area pellucida and peripheral area opaca. Area pellucida is a transparent because it overlies the blastocele, whereas the area opaca is opaque because it overlies the yolk. Anteriorly, area pellucida has a temporary mesoderm free area called proamnion. So, proamnion is a mesoderm free area. In the middle of the area pellucida is a primitive streak. It has a central canal called primitive groove. On the either side of the primitive groove, there are primitive ridges. Primitive groove ends in depression called a primitive pit. Anterior to primitive pit, there is a thickening of cell called Ensign's node. So, Ensign's node is a thickening, particularly in the anterior to primitive pit. Anterior to the Ensign's node is the head process with a centrally located notochord. The diagram gives certain parts like proamnion, notochord, Ensign's node, primitive streak, area opaca, area pellucida, embryonal area, neural plate, anterior border of mesoderm. Anteriorly there is a cephalic end, posteriorly there is a caudal end. This is something about 18 hours chick embryo. Second one is 24 hours chick embryo. The whole mount of 24 hours chick embryo. Here, the embryo shows two distinct regions, a central clear transparent area pellucida and a peripheral opaque area opaca. Area opaca is again differentiated into an outer area vitali and an inner area vasculosa. Area vasculosa contains blood islands. Cephalic regions undergoes a rapid growth and it extends anteriorly overhanging the proamnion. The cephalic region projects free from blastoderm may now properly be termed as the head of the embryo. The space between the head of the embryo and blastoderm is called the subcephalic pocket. Notochord lies in the midline below the neural plate. Neural plate is prominent and the neural groove is deeper near the cephalic end. Neural fold appear as dark bands. There are four pairs of somites are seen in the midline on the either side of notochord. Unsegmented mesoderm is seen behind the somites. Primitive streak is reduced and restricted to the posterior region of area pellucida. Anterior to primitive streak is the Ensign's node. So the diagram gives certain parts like ectoderm of head, border of foregut, subcephalic pocket, margin of anterior intestinal portal, area pellucida, unsegmented mesoderm, blood island, area opaca vitellina, proamnion, mesenchyme, neural fold, neural groove, notochord, somites, Jensen's node, primitive streak, border of mesoderm. This is something about a whole mount of 24 hours chick embryo. Next one is 36 hours chick embryo. The whole mount of 36 hours chick embryo gives certain peculiarities. The brain is differentiated into forebrain, midbrain, and hindbrain. The optic vesicles are established as paired lateral outgrowth of the forebrain. There are 13 pairs of somites are formed. The primitive streak becomes shorter because of the lengthening of neural tubes. Middle region of the heart is considerably dilated and bent to the vitellin. Proamnion, notochord, neural groove, neural fold, area vasculosa, area pellucida, and Ensign's loop are present. The diagram shows certain parts like forebrain, optic vesicles, proamnion, notochord, neural groove, neural fold, area vasculosa, lateral body fold, vitellin artery, area pellucida, Ensign's node, primitive streak, mesoderm, 13 somites, first somite, vitellin vein, hindbrain, midbrain, foregut. This is something about a whole mount of 36 hours chick embryo. Next one now, we'll discuss something about 48 hours chick embryo. The embryo appears like a question mark. It is one of the basic point to identify the 48 hours chick embryo. It has a question mark shape there. Cranial and cervical features are distinct. Head and neck of the embryo are twisted due to the torsion effect, so that left side touches the yolk and the right side away from the yolk. So, head and neck of the embryo are twisted due to the torsion. So, it looks like a question mark. Chambered mature of the heart distinct. So heart consists of matured chambers, distinct showing the ventricle, atrium, bulbous, arteriosus, and sinus venosus. Presence of about 28 pairs of somites. There are 28 pairs of somites. Brain is differentiated into mesencephalon, metencephalon, prosencephalon. Vitelline artery, vitelline vein are well developed. Presence of tail, the presence of tail bud and hind limb bud. Presence of auditory vesicles or optic vesicles. Primitive streak is in the form of primitive plate. So the diagram conserves certain parts like prosencephalon, mesencephalon, metencephalon, myelencephalon, and then optic vesicles, margin of head fold of omnion, truncus arteriosus, ventricular region, arterial region, and then ophalomesenteric vein, extra embryonic vascular plexus, ophalomesenteric artery, neural tube, primitive plate, unsegmented dorsal mesoderm, lateral body fold, lateral mesoderm, somites, sinus region, auditory pit. These are the few important parts there in the whole mount of 48 hours chick embryo. Later I will discuss something about the TS of 18 hours chick embryo and TS of 24 hours chick embryo. Any clarification, any doubts, send me the messages. Thank you.